So I grabbed a bunch of dead bees from the colony. I'm going to do a quick Nosema check. What I'll do is I'll pick 10 of these bees randomly. And then I'll do a couple of individual bees just to give me an idea of what's in their stomachs. So I'll pick a tiny bee for the individual bee. So I'll put this bee here, pretty small abdomen. I'll squish it on that slide. And then I'll find the biggest bee in the group here. And then I'll put that on the slide too. So let's see. I'll just take, let's see this one. See, they're all pretty tiny. Yeah. So let's see. I'll take this one. It's got a bigger abdomen. And I'll put that one over here. And then what I'll do is I'll take 10 of these bees and I'm going to squish them as a whole. So it'll give me a general idea of Nosema. So 1, 2, 3, 4, 5, 6, 7, 8, 9, and let's go with 10. I'll just put these on the side because they're thawing out. Don't want to leave bee juice everywhere. And I'll clean that up after. So what we'll do is we'll put 1 ml per bee inside the packet here, and then I'm going to squish them together. So that would give me, so if there's 10 bees, so I need about 10 mils. So let's do that. So this is just distilled water. Okay. Oh, so there's a bit more than I need. Just use this syringe and suck it up. Go. So there's about 10. And then I'll just squeeze it in there. And then we're going to macerate that. With the water. My little home lab isn't the biggest, so I have to be careful not to knock things over. So easiest thing to do here is just use a mason jar. I've got a honey jar here from my other project, so I'm just gonna use it to squish all these bees. And then I'm gonna basically, there we go. So you want all the contents out. And then I'm gonna use my fingers just to make sure that everything gets well mixed together. And then I'm gonna take a drop of that, put it on the slide, then put a cover slip. And that's that. So you can see over here there's some feces there stuck together, so it's important to mix everything together. And the reason you put water in there is to help with the mixing, and also here we go, just want everything to be in the corner. And then when the corner, you can just squish it like that. Just mix it, and then I'm going to use a little chopstick there. You can use almost anything, just to put a dab of the liquid on top of the slide. Let's see, I've got a mixing stick here. Okay, so just keep mixing it. Again, Nosema spores are kind of heavy, so they'll sink. So you want to make sure you don't wait too long after maceration to take your little sample. What I'm going to do is just mix it around. Okay, like that, and then put a drop. I'll put a couple drops like that, and that should be good. And I'll put that on the side. Close this up so it doesn't leak out, and we'll clean it up after. So what we can do is just take a slip. I'll need a couple, so we'll take one to start off. And I'll just put it on there. Good. There's one. And I'm just going to use the outside edge of this, these tweezers to pretty much squeeze the guts out. So I'll grab her by the abdomen, and then you just push down. And then you push her poo out. You see it's fairly dry. It's a very concentrated feces, which is normal. So maybe these bees actually died of, I'll say, of dehydration. So I'll just add a couple of drops of water on there just to help mix it. Yeah, so I'm just going to use this end to mix it around, to break it down a bit. Before I put this lip on, you can see it's much more concentrated than the first sample. So this was the big B, so I'll write big B on this stick. Okay, so I'll put my slip on there, and then we'll do the last one. Okay, so it's important to keep decent records. So I've got a Sharpie here. I'll just put 10 on this one for the 10 B's. This one I'll put B for big, and that one I'll be small. Okay, so there's 2 slides, and then one more to prepare. I'll actually use a different pair of tweezers this time. Don't want to cross-contaminate. And this is the small bee, and again, you're just going to take it. You could use a butter knife to do the same thing. You can see her stomach's actually empty. Nothing in there. Nothing's coming out. So they might actually be dehydration. So these bees are from my biggest double colony, and there was actually quite a few dead bees on the bottom board that I scraped up. So yeah, let's try again, and yeah, let's see if anything comes out this time. I get very concentrated poops. So I'm doing another little project now on water balance in the colony. So one way that the bees actually moderate or control water in the colonies is through basically their system. They're able to recycle water, digested water, so through their system. So that's one way they do it. But yeah, so let's see what's in this poo. Get another slip. I think there's only one. Oh, there was 2. And I'll close that up. There we go. And I'll push it in the middle. Wipe the edge a bit. Oops, scheiße. I'm making a mess now. I put too much water. Okay, so you can see nothing's perfect. Just wipe the underside of this. And you don't want to contaminate your microscope either, so I'll just clean the edges here. It is for a quick check, so it should be okay. It's drying up already. So let's go have a look in the microscope. So I'll stop the camera. Okay, so this is TubeView. This is the software I use for my microscope. So I've got those 3 slides ready. So what I'll do is I'll start with the 10 bee gut sample. Just to have a quick look, I'll start at 10 power, just to get it in focus so I can have a quick look. Usually you won't be able to see much at this level. So yeah, first thing I see is those amoebas. I'm assuming, let's see. Yeah, so I'll just darken it a bit. So you'll notice these white globes. It's another Amoeba cysts, it's another disease I'm looking at. So it potentially has an effect on, again, the it's in the digestive system. So there's quite a few of these globes. What I'll do is I'll go to 10 power. Really dark, so I'll have to lighten it up and get it into focus. And I do notice a bit of nosema at first glance. So you can see how these oval shaped items, I guess things, those are Nosema spores. There's not a lot, so it's not too worried about Nosema levels. They are dead bees outside the hive. But again, you can see these white globes now. So I will take a few pictures because I'm not an expert at this. I'd like to get a proper diagnosis or somebody to confirm what these things are. So there's a bit of Nosema with, for example, in this slide or this view, there's 1, 2, 3 of those globes, and there's maybe 4, 5, 6 Nosema spores. Typically, if I showed you a high concentration, this here would be just full of Nosema spores. So not too worried about it yet. Okay, looks like pollen, some more pollen. Those globes again, it's still some tension in that slide. More globes. So just for the camera's sake, so right now I'm at ×40, so magnification. So I do have a measurement tool on the device, so it's all whatchamacallit, it is calibrated. So what I'm going to do is I'm just going to take a snapshot and I'm going to do a couple of measurements. So I'll do that, snapshot, and let's see, snap. Okay, here's the first picture. So it's got about 7 micron diameter. This one's a bit longer, but generally the same size. Get the 2 axes. Oops, delete that. So let's see, delete that one. So again, if I measured the Nosema spores, enable it. I'll just go back to the live camera. Okay, just want to check to make sure that calibration works in the picture too. So yeah, I get the same 705, which is good. And then the Nosema spore here, for example, boom. I'll just go back to the picture and I'll zoom in, oops, to 33%. And let's see, let's just do one that's fairly elongated. So I'll take it from end to end. It's about 5. Like that by 3. So I think they're typically in the 6 range, 6 microns. So ballpark okay. I'll confirm that and just make sure there, but it pretty much looks like Nosema. Okay, I did a bit of research on the Amoeba disease. So it's a digestive system disease. It's caused by a protozoan called this. And basically there are some relations that spread similar to Nosema. Usually doesn't cause mortality, but but it impairs the functioning of these Malpighian tubules, which is like the kidneys of a honeybee. So it's diagnosed via microscopy, and I guess in the past I've noticed it when I had Nosema and high mortality. So I guess in the cold, when the bees are stuck in a colony for months on end, it probably has a bigger impact than in areas where bees can cleanse. Just from the looks, so what I measured around 7 microns, and it does have this general shape, and it's got these things inside the cell. So I would say with a fairly strong sense of certainty that's what we've noticed, those globes. Let's see what else. So a bit on these tubules. So I got this from American Bee Journal. So basically, it's spaghetti-like. It floats inside the body cavity. It's connected to the midgut, and it's part of the water system. So the osmoregulation. So it does produce uric acid, so urine within the bees. So if this ain't working properly, then the whole osmoregulation of those individual bees that do have this will be impacted, which is a big problem in winter, especially if there's no cleanse. And yeah, not sure if it's connected to those guts being so dry, but to be determined. So fairly similar explanation from an older paper back in '79. So just about it being used to flush nitrogen waste, so uric acid into the hindgut, but also it's used for reabsorption. So any moisture or water contained in the gut can be reabsorbed into the body, and it's a way of conserving moisture, especially when they're clustering and then in cold winter environments. Pollen. So I won't do a pollen class now, but that there's probably goldenrod, the native goldenrod. So technically what I would do is, it's got all these spikes coming out, so it's echinate. And asters are the types of pollens with echinate. Yeah, so it's about 20 microns wide. So that pretty much aligns with goldenrod, the northern type. So this here you can see you can sort of see this surface texture. So it looks like a brain, and it's got a pore and a furrow here. So there's so when you're doing pollen analysis there, you're going through different zooms to get different textures to give you an idea. So like for example, this one here has more of a striation type surface. I'm not exactly sure what that one is, because yeah, typically I'll put them through an alcohol bath first when I do check microscopes or pollens to give me an idea of what they are. So for example, this thing here, this is a fungal spore. It's common in honeydews, but also I've found it in pollen balls. So basically it's a lot of these spores are wind transported. So you'll actually catch them in the air. So it's fairly common. So yeah, the few Nosema spores, nothing too crazy. Remember, this is for 10 bees, so it's not just a single bee. So you divide this concentration by 10 bees, and it doesn't look like any one bee is very, very sick. I guess the thing I keep seeing is these globes. So, and so just so you know, anytime you're looking at your telescope or microscope and you see pollen grains with these spikes, you can pretty much assume that it's a naster. So dandelions, yarrow, goldenrod, arnica, all those types of things will have these spikes. There'll be different sizes and they'll have different sections. So again, there's another globe up here. Again, I won't assume what they are, but they might be an amoeba cyst. And there's a square shape. It's probably a sugar grain. So what I'll do is I'll actually go to the next slide. And let's have a look at something that's more concentrated. Okay, it's a whole world in here, so it's an interesting world. The bee gut. Okay, put it back to 10, take the slide off, and I'll start with the big bee. And you can see right away, a lot more content. I just use a couple drops. So a lot of pollen grains and broken down grains, which is normal. At this magnification 10, you'll see, I'll give you a general idea. So looking, I see some fireweed, because fireweed's really big. So let's see if I can see. Do you see the fireweed? So this grain here is most likely fireweed. So I'll put it on magnification 10, and then I'll just give it a quick measurement. You can see in the 60 range, and yeah, that's 70 range microns. So yeah, those are fireweed pollens. So I'll zoom around a bit more. But you can see this bee is eating and going through things even in winter. There's a mega pollen. Not sure what that is. This I do feed pollen patties, so there might be some foreign pollens that I don't know about. So it's massive at 110 microns. Interesting. So I'll zoom into it. I'll lighten it up, so add more light. I've got to change you can see how it's got these little pores, Interesting. So I'll change my magnification back to 40. You can see how it changed the measurement. So I'm just curious, so I'm going to actually take a snapshot of this, see if I can figure out what it is. So delete this because I need a measurement. Let's try to get the edges sharpened so I can get a true width. There we go, so about there. So measurement. It's 120 microns. Interesting. Good. Let's see. So I'll actually leave it on there. So 122 in the other direction. And now I'm going to just show the surface texture. I'm going to take a snapshot. So you can see these little, interesting. I've never seen anything this big before. So let's do, we'll use a snapshot, see if I can get different textures to come out. Yeah, so it's a netunate with really interesting. So my pollen book's not at home right now. Somebody's lent it out or borrowed it. Okay, let's keep going. I'll delete that. Okay, so let's zoom around a bit. So I'll check the edges. Oh, interesting. So this one here, for example, this is a probably lingonberry. So the lowbush cranberry grows everywhere. So this is a June, early June, mid-June pollen, and let's see, yeah, more early June. So you can see it's in their stomach, so they probably had some stored or some honey stored from earlier in the season. So again, you can see some of these smaller asters. So if I measure that, and I'd say it's probably important if you do have a microscope to get a calibration tool, especially if you have a camera. So it's about 20. So again, it's likely the aster. Oh, it is an aster, but it's probably that decumbent, the northern goldenrod. So I'll check the edges. So one thing I don't see, there's another big monster one. I don't see any Nosema, which is good. So there we go. So there's one here. Oh, I'll center that. So see, again, it's hard to see, but this one has spikes also. But you can sort of see like a shape on the side here. So these spikes, these echinate, so this is an aster, but this is likely, so again, so about 30, let's do this direction, yeah, 25. So this one here is likely, I think it's a hawkweed. Dandelion is slightly bigger than that. Lots of big stuff in there. Interesting mix. So again, there's another lingonberry in the middle there. You can see how I focus in and out. So it's, for me, it's to help me see the textures and the pretty much the morphology, the shape of these pollen grains. And it's neat to see pollens from beginning in the season. So it means that that they are storing a couple of rust spores. They're storing and eating material from May and June in the winter. So anyways, it's an interesting bee gut. I'll probably put this on a separate video because I'm talking long down in the bottom there. So this thing here, over here, this is looks more like a rust spore. And yeah, I'd say generally, oh, there we go. Sort of hidden in the background here, there's a big fireweed, pollen. And yeah, because it's been in their bee gut, so it's being digested. So you can see how it probably the acids in the stomach chew up and break down some of these pollen grains, so the textures will be different. More fireweed. head over lingonberry. Interesting. There's another fireweed. That's half digested. Some rust spores. Okay. Probably just an air bubble. There might be a couple little Nosema spores mixed in there, but I don't really see them. Usually really dark brown material, like in the bottom there. That's typically fungal spores. Okay, so I'll do one last check. I know I'm moving around quite a bit. Okay, so let's see. Some more fireweed, and then it gets too concentrated. That's a challenge with concentrated feces, is it's too dark and you can't see through it, so you have to add a bit of water. There's another one of those big ones. Interesting. We'll have, I'm guessing it's from the pollen patties. Rust spore. Okay, and then let's do the last slide. Okay, just center it. Again, it's too bright, so I'll darken it up a bit. Get it zoomed in. There we go. See how it's got, it's less concentrated, so it's more spread out. And again, we'll go around. There's that big one again. Keeps popping up. So let's zoom in until I want a picture of it. There we go. It's centered. I'll go to 40. Okay, so there we go. Let's take a couple pictures of that. There's the itchinate, so you can see how it's these little spikes coming out. So all these little circles here is actually one of these spikes. So I'll just get to use the snapshot tool. Before I do that, I'll actually put I'll put a 10 micron scale on it. Okay, let's try that again. Capture, and then I'll do another measurement. I'll just get the edges defined, and let's do that. So let's go from here to there, so it's over 100, it's about 115, so it looks to be, it's got 100 to 120 microns diameter. Cool. So let's zoom out again. Let's go have a look around. Okay. I'll just zoom in a bit to see if I can see some Nosema spores. At this magnification, you won't be able to see much, so I'll see how bright it is. So if you're going to use times 10. You just want to darken the screen a bit, so less light is a bit better. You'll get sharper contrast. Okay, so I'm just going to line it up a bit. And Nosema spores are heavier, so they tend to sink. So when you're scanning through your slides, you want to check different levels. So let's see how. Okay, so at all levels it seems fine. And these are just broken down pollen particles and stuff that they ate. So again, there's some pollen, some rust spores, some bee hair. Some dark stuff. There's some, that brown again, it's right here. These are fungal spores. There's lots of different kinds. Don't ask me what they are. Oh, there are the globes again. So let's zoom in and out. There we go. So there's a few of these globes. So this was a smallish bee, so I had to redo it, but you can see there's some globes here. And again, it's, I think those, it's an Amoeba disease there. There's not much research on it, but there is a link with one of the tubes there, the mag tube. I can't pronounce it again. So, and it probably, there's a connection with dysentery and Amoeba disease, and mortality if it's associated with Nosema. And yeah, there's quite a few of these globes in this one here. Interesting. Good. So again, you can see that these globes are smaller. Nosema spores you can see at times 10. So it's important to basically magnify down to a higher magnification, just so you can actually see stuff. So again, these globes, they are quite a bit everywhere. And yeah, so I'll try to figure out what they are. Cheers.